Okay, so today we're going to be focused on the immune system. Today's video is going to focus on evolution and the innate portion of the immune system, and then next video will focus on the adaptive. So hopefully most of you guys know and remember that your immune system, its job is to protect the organism from pathogens. Okay, so pathogens, in case you guys forgot, pathogens are anything that can cause disease. Okay, so disease is basically anything that will disrupt homeostasis. So your immune system's job is to act as a line of defense to protect you from these pathogens and foreign invaders. And it has two basic divisions. Okay, it has the innate, which is non-specific. Like I said, we're going to focus on that a lot at the end of this. But these are things like your skin, things that act as barriers, your general chemical and cellular responses. But again, they're non-specific. So you're going to find these in most of your multicellular organisms. So plants have these, animals have them, fungus have them, insects have a innate immune system. So most of your multicellular organisms will have this. Your adaptive immune system is a specific response. This is specific antibodies that are made in response to a specific pathogens. Talking about B and T lymphocytes. Okay? So this is a slower response than innate, but again, it's more specific. And this is only in jawed vertebrates. Okay? So only in organisms that have a jaw as well as a backbone. Okay? And so based on that information, hopefully, because like I said, we're going to be talking about evolution in this video, hopefully you will realize that the innate immune system is going to be older. Okay? It is thought to be what's considered an ancient, conserved biochemical pathway. Okay, hold on just a second. Okay, so you have it there. Your innate immune system is thought to be an ancient, conserved biochemical pathway when compared to the adaptive. Which should hopefully make you realize that the innate immune system is evolutionary older. It's going to be your dominant immune system in plants, fungus, insects, primitive multicellular organisms. Okay, because your adaptive immune response had to have come about later in the evolutionary line. Okay, and we'll look later at some similarities between plant responses and animal responses to kind of show how this immune response is very similar in the various types of organisms. Okay, so what we have here would be a macrophage, that's this big cell right here, okay, that is eating some of these bacterial cells. So let's look at these pathogens and look at why does the pathogen invade in the first place? How come the pathogen came into your body in the first place? Okay, so it could be looking for water or food. Okay, so it's looking for almost like a host. So looking for a source of food or water. It could be seeking protection. Okay, finding somewhere where it will be protected from maybe coming inside your body to be protected from the outside environment. It may need to reproduce, and so it needs to find a place that is suitable for reproduction for it. Okay, and so your defense mechanisms have evolved to help get rid of these pathogens. You're trying to maintain homeostasis by removing these pathogens that are disrupting your homeostasis. Remember, your homeostasis is that constant internal environment. Okay, so the immune system is thought to have evolved from amoebas. Okay, and so if y'all don't remember what an amoeba is a type of protist. So it's a eukaryotic cell, but it's an amoeba does phagocytosis. Okay, so here's your amoeba up here. And for an amoeba to get food, it's going to do phagocytosis. And remember, that is cell eating. So it's going to reach out. Okay, it's going to grab whatever it wants to eat and bring it inside and then digest it inside. Okay, and so we've got some cells in our immune system that work in a very similar manner. And one of the theory is that the thought process is that it would be advantageous for the amoeba to be able to recognize self. Okay, so that as these amoebas, they're freshwater organisms, and so as they're floating around in the water, when they bump into another amoeba, hopefully they don't eat it. Okay, so they want to be able to recognize self versus non-self. And they would do this by receptors. right? There are what are called signature molecules. Okay, and so signature molecules are specific to a particular organism. Okay, so signature molecules, they act as identification molecules, and they're unique to a particular group of organisms. Okay, like an example would be cellulose for plants. Okay, when something encounters an organism and that organism has cellulose in it, then it's automatically known that that would be a plant. Okay, so the thought process then was that the amoebas would have an interaction between their receptors and these signature molecules, and that's what would tell them whether it was in another amoeba or not. And it'd be a form of cell communication, right? If we've got our receptor, which if the receptor is triggered, when it comes in contact with the molecule or with the, the other amoeba. So when the receptor is triggered, you guys remember this? So we trigger the receptor. So that's my reception part of my cellular communication. That would trigger a pathway inside of the amoeba that would result in phagocytosis. There'd be my response, would be phagocytosis if the cell attached. If it was not an amoeba cell and it attached to the receptor. Okay. So this pathway has been conserved and passed on over time. You can see here in plants, okay, we've got the pathogen or the PAMP. We'll talk about what a PAMP is in just a second. Okay, eliciting the pathway and then the immune response. Same here. Okay, we've got our signal coming into our receptor, triggering a pathway, which triggers an immune response. Okay, so this basic pathway has been conserved and passed down through time. The differences would be due to mutations. Okay, and so as the mutations occur, the pathway changes slightly from organism to organism. And so those PAMPs that were mentioned here, okay, those are going to be what are called pathogen-assisted molecular patterns. Okay, so pathogen-assisted molecular patterns. And so these PAMPs, they act as signature molecules. Remember those molecules that are specific to a particular organism. So they act as a signature molecule or an identification type molecule. So the PAMP can act as a signal to that receptor to let it know that maybe it should cause an immune response to happen. So let's take a little bit closer look at plants here. Okay, so we're going to look a little bit closer now at plants. So plants, like I said, they have a form of innate immunity. So this can be a non-specific immunity. And so the first one would be physical barriers. Okay, so we're looking at things that would physically prevent something from harming or getting into the plant. Things like thorns, thorns, hairs. You can see all these hairs on this flower here that are too close together. And so the insect can't actually get in there to penetrate. Obviously, thorns and spines, the sharpness would keep organisms away, hopefully from eating them. The plant could also have a very waxy cuticle. Okay, remember the cuticle is that kind of waterproof covering on the surface of the leaf. So it can have a very waxy cuticle that the bacteria or the fungus cannot penetrate. Like if you think about like a succulent and more of a desert type plant, a very thick waxy cuticle, and the bacteria, the fungus wouldn't be able to penetrate that. So those would all be examples of innate immunity, non-specific immunity. Okay, let's take a look now at these secondary metabolites. Remember, everything the plant does is going to be non-specific. Okay, so let's look at these secondary metabolites and what those can do for a plant. Okay, so secondary metabolites are found in plants, and they are metabolites that are not necessary for plant life. Okay, they are not required for plant life. They have an alternative function. Okay, they have some other use or function. They will harm, as a general rule, they're going to harm the herbivore in some way. They're going to have an impact on the herbivore, usually to discourage the herbivore from eating the plant. So these secondary metabolites, like you said, they act on the herbivore. They could impair the development of the larvae. Okay, so the babies that are laid could have some kind of developmental issue. Okay, they could damage the digestive tract of the herbivore that eats it. They could also act as a fungicide. Okay, remember side means to kill. On the last slide we were talking about side as well. That means to kill. Homicide kills people. Okay, so they could act as a fungicide. So there's a variety of functions that these secondary metabolites could have. Let's take a look at one. We always like to have a good example. An example is always good to use when you guys are answering your free response questions. So the one on the right down here, the canavanine, is produced by the plant. So that's the secondary metabolite. And it's very similar to arginine. Arginine is an amino acid that is required. Okay, and if you look, here's your only difference. Okay, doesn't seem like a very big of a deal. Okay, the tRNA can't distinguish between the two of them. So when tRNA is looking for an arginine to pick up, it can pick up this canavanine that the plant has produced. Okay, so the tRNA can't distinguish between the two, so it can be substituted. And so when protein synthesis is happening and it calls for arginine, okay, the tRNA may bring over this canavanine. And what that will do is it will affect protein structure, and it will result in an inactive protein, a non-functional protein, that can actually end up resulting in death of the organism. So some of these plants and herbivores have actually evolved together as a form of coevolution. The example that we have here is the butterfly and the passion vine. Okay, and so what happens with the butterfly and the passion vine is that the secondary metabolite that the passion vine makes, the butterfly has been able to evolve so that it can break down that secondary metabolite. And it actually uses the nitrogen from that secondary metabolite. It uses that nitrogen byproduct. So the two have evolved together. As the passion vine has developed the secondary metabolite, the butterfly has developed the ability to break that down and to be able to then actually use it. So let's look at another defense of plants. So this is another innate defense, but it's called it's a type of induced defense. Okay, so something has caused this response. And so what happens with this type of induced defense, okay, it's called a hypersensitive response. Okay, so we have this hypersensitive response, or HR response, that will happen. And the result is going to be the very quick localized death of the plant cells. You can see all the brown dead spots on this plant. Okay, and that will happen very quickly in response to the pathogen. Okay, and so these are going to be done in response to what's called an virulent pathogen. Okay, and most of the pathogens, definitely in plants, are going to be this way. These are pathogens that cause disease, but they do not cause death. Kind of like how a parasite does not necessarily want to kill its host, because then it's got to go find a new host. And so what happens is when this pathogen invades, okay, it produces what's called the avirulent pathogen invades. It triggers the receptor here. Okay, and that receptor triggers your signal transduction pathway. And that's going to end up causing the plant to produce what's called R protein. So the avirulent protein that's made by the pathogen is recognized by the R protein in the plant. And so this R protein recognizes it, and it triggers the response. Okay, and so the response can have a variety of outcomes. Okay, it can end up sealing off the area, so that it hopefully keeps the pathogen contained and it can't go anywhere. Okay, it can produce fungicidal or bactericidal, remember, things that can kill molecules. Okay, it could also produce hydrogen peroxide or nitric oxide. Okay, so it could produce hydrogen peroxide or nitric oxide, which are both toxic to cells. So that would help in the localized death of the cell. It can also produce what's called methyl salicylic acid, which will trigger a systemic acquired resistance. So we'll look at that one in just a second. Okay, but again, so these avirulent pathogens, the ones that do not necessarily kill, okay, they cause disease but not death, they trigger this hypersensitive response. So again, another form of cell communication happening, which is going to be that very quick localized death of the area of the plant cells. And then this can trigger an even further response called SAR. So let's take a look at that one real quick. Okay, so remember this hypersensitive response here, okay, that has triggered the production